Okay, good afternoon. This is Mr. McGee. I'm going to show you how to do our lab, and this is on genome size analysis here. You're going to need to click this database here to get your information, but this untitled document is going to be where you're going to put everything. On this untitled document, you can see what we're asking here is a research question. Always start with a research question. Is there a correlation between the genome size and the amount of DNA? Or we could ask, is there a correlation between the amount of DNA, the number of base pairs, or the number of genes? What we're really wanting to find out from this is if you have more DNA, does that translate to having more genes? Or if you have more genes, do you necessarily have more DNA? Can we predict it? And what degree can we predict this? You're going to need to calculate what's called a correlation coefficient to also determine this. So first, we're going to need to make some graphs. Then we're going to calculate the correlation coefficient. You will make three separate graphs. One of them is going to include prokaryotes and eukaryotes. One of them is just going to be on prokaryotes, and the other is going to be just on eukaryotes. Okay? That's pretty much it. And let me go ahead and get us started with that. You're going to need to open your database here, and I'm going to blow this up here a little bit just so it's easier to see. You'll notice we have all these different specimens, and they're alphabetized by the species name. We also have the number of base pairs. If I move this down, it might be a little easier to see. Number of base pairs in the genome, and this is the number of protein coding genes. So this organism here has 1.7, roughly, 1.7 million ATCG sequences. But they have a total of 1,800 genes, well, almost 1,900. Humans, I highlighted here so you can see it a little easier, have 3.3 billion ATC base pairs in their DNA. That makes a total of 21,000 genes. But again, is there a relationship? If you get more DNA, does that necessarily mean you're going to have more genes? So that's what we're going to try to find out from this. First, what we're going to want to do, because according to our assignment, we want to do prokaryotes and eukaryotes, we're going to want to sort these according to the prokaryotes and eukaryotes. Click up here, species, drag and highlight all the way down into this corner there. And I'm going to need the menu here, I guess, so you can see this. But you're going to highlight everything, and notice we have our headings here of species, base pairs, protein, etc. You're going to go up here to data and sort range. This will allow you to say, yes, my data has a header row, and we can now sort according to any one of these. It's already sorted by species, but I want to sort it by the domain, because that is prokaryotes and eukaryotes. Let's go ahead and sort it. And there we go. I have all the eukaryotes alphabetized on the top with the prokaryotes below it. So now what we're going to want to do is we're going to want to create some graphs. I'm going to go ahead and just create, let's go ahead and do the bottom one here. I'm going to answer the bottom one. The other two are the same thing. You just got to do it for their specific reason here. Okay? What we're going to want to do here now is make a graph of the base pairs and the protein coding genes. To do this, there's a couple ways, but the easiest way is click on the data for the number of base pairs and highlight it. Make sure you get all of the eukaryote data there. And then what you're going to want to do is highlight the next column here, but if you notice, if I don't hold anything, it's going to unclear that last column, and that's a problem. In order to get it to let you highlight multiple columns, you need to highlight one column, and now click and hold Control on your keyboard. On a MacBook, I'm not sure if you use Command or something. I'm just using a Windows PC, but click and hold Control, and now highlight the second column here. Once we've done that, you can see that essentially what we've done is the computer has identified this as the x-axis and this as the y-axis of your graph. So now that we've identified the x and y-axis, we can just go up here to Insert Chart, or there's an icon here which is the chart insert icon. Either one works. Okay? And it makes us a bar graph. However, as I've said in class, a bar graph is only used when you have categorical data. So if we were using these categories of species, yeah, we would use a bar graph. But we're using numbers here, and numbers change in different increments. And so we don't want this. We want to have a scatter plot. So go over here to Chart Editor Setup. By the way, if this ever closes, you can just double click on your graph and it'll pop back open like this. Okay? Let's go back here to Chart Setup, and you're going to go to Column Chart and go near the bottom and pick the scatter plot. Okay? So we could go ahead and just copy this graph and paste it right now, but notice it says you have to make a logarithmic scale. So to do this, I'm going to go here to Customize, and now I'm going to want to go down to Horizontal and Vertical Axis. I'm just going to do the horizontal axis here, but again, a axis here, if I can, whoops, go back on my slide for this. We first want to look at what is a logarithmic scale. This is the Dow Jones Industrial Average of the stock market. You may recall me sharing with you in class that this scale here at the top, it's going up in even increments from 0 to 5,000, 10,000, 15,000 would be at the top. And you can analyze it like this, but the problem is it doesn't show us the rate of change and it doesn't handle exponential growth very well. And so it's very deceiving when you look here and you see the stock market crash in 1929. It doesn't look very bad, and this one looks much worse, but the reality is more apparent when you use a logarithmic scale. A logarithmic scale is when we increase our axes at an exponential rate. In this case, it goes from 10, 100, 1,000, 10,000, and you have to account for this when you're looking at your data. Now it's more apparent that the rate of change and the amount of change was much more severe in the stock market crash of 1929 than it was here at these points in time. But you still need to account for this when you're using logarithmic scales. Both of these can work, but that's why you want to use different scales for different situations. Okay? In this case, we're going to go back here to Customize, and we're going to change our horizontal axis here to logarithmic because it's right now linear, going up 1 million, 2 million, 3 million. Let's go ahead and knock this one here. We're going to go horizontal axis and change it to a log scale or logarithmic. Notice what it did there. Instead of being a nice even increment, it's now going up exponentially from millions here all the way to billions all the way over there. And so you can see what a logarithmic scale does. It's not changing your data, but it's changing the scale of the axis. Okay? Now, I don't think we need to change the vertical axis here because it's not accelerating, or the number of genes is not accelerating as the number of base pairs is accelerating. Okay? So we can keep it like this, and you could probably play around and tweak with this if you would like to. So why don't you do this if you get some time here. Go under Customize, play around with the chart titles, play around with your, series legends. Just get used to this because we will be using this more throughout this course, okay? You can copy and paste this graph here, and you paste it right below there like this. Actually, I'll just go ahead and copy and paste it. Okay, here is my graph, and I'm going to bump all this down here just so it makes it a little bit easier for us to see. Okay. So here is my graph, and you can see I just need to calculate my R-square value. And if I go back here, just like I showed in class, R-square is a measure of how related two variables are. An R-square of 1 means it is a perfect correlation. All the data points fall along a predictable path, whereas an R-square of 0 means the data is all over the place. Anything that's above 0.7 is usually a strong, and anything below 0.3 is considered a very weak correlation. Okay, so we want R-square because it tells us how correlated is our data. When we look at the weathermen, for example, when they often predict the weather and then we see the actual weather, they're usually pretty close, but they're not, almost never I could think of where they're exact dead on in their predictions. We would say they have a high R-square, therefore, but they're not perfect, and the correlation between their predicted and their actual weather. What we want to do here is see what is the R-square of this. Now, if you look at the data, you might be tempted to think this has a high R-square because all the data seems to be falling along a line, and that's where this gets tricky. Because while it does seem like it has a high R-square, remember, if we go back to the horizontal axis here, and I change my log scale, there really is no correlation in there that is apparent. And so this logarithmic scale, once again, you have to learn how to read and interpret these. It's misleading to think that this shows a correlation. It might be correlated early on when we're under 100 million base pairs, but beyond that, these outliers are really throwing our data off there. So let's go ahead and calculate the R-square value. I'm going to clear that off there. We're going to need these two columns. To calculate R-square, you just click anywhere you want a calculation to be made. hopefully you pick an empty cell somewhere, and you click in equals, and now you can enter any equation you want to. But for r squared, you're going to calculate RSQ. It's automatically known by Google Sheets and Microsoft Excel. And you can click on that. Equals RSQ for r squared. And if you're confused, what am I supposed to enter? You can click the question mark. It says you're going to enter the data for y and the data for x. Okay, that means it wants our y-axis separated by a comma with the x-axis. Let's go up here and try that. I'm going to enter my y data first, and now I'm going to put a comma, and let's enter our x data, and now hit a comma, or whoops, we don't need that comma. We just need it to separate. And now hit enter. I got an r squared value of 0.108. And you can ask yourself, is that a high correlation? 0.108. Well, I will let you interpret what that means. Okay? But again, it's the logarithmic curve that may make that deceiving. One last thing we need to look at here is rounding it to the proper number of sig figs. If you recall, the rule is you look at your raw data that was used to calculate this, and you round it to one more sig fig than your lowest value. So this here is a 9. We're going to want to, and it's only one sig fig, so we're going to want to round this to 2 sig figs. However, I did not use that 9 in my calculation. I used these numbers here in the calculation. So let's go look and see what kind of sig figs I have here. If I go through and look, I see several places where I have 2 sig figs here, 17 and 21, 23,000 and 2.3 billion. So I have 2 sig figs that is bare minimum, and that means I'm going to round it to 3 sig figs, one more than my lowest number of sig figs. 108 is the answer, 0.108. Come up here, enter 0.108, and you are done. It's a low correlation. Okay, with that said, you then come to this one, do the same thing for the prokaryotes, and then both of them. If you have any questions, please let me know. Otherwise, good luck to you all. And please make sure you play around with the chart editor so you get used to how to use this properly.